Welcome back, friends. In the previous session now we discussed about the phytohormones and its characteristics, and we saw how the phytohormones they are differing from the animal hormones. And as a part of recap, we said that the plant hormones they are chemicals which are required in small amount as how animal hormones they are, and they influence growth, development and differentiation of cells and tissues. So in most of cases the plant hormones they tend to stimulate growth. However, some of the hormones such as abscisic acid, they are working antagonistically, basing with the working mechanism of other hormones such as auxins. So if we are comparing the working of auxins and abscisic acid tend to promote abscission, but we will discuss it in detail later. Now, in the second characteristics, they are often produced and used on local basis within the plant body. This is different from the animal hormones, what they are doing. In the animal hormones, we know that the endocrine hormones they are produced in some of the regions of the body known as glands, and they are transported by blood to different sites where they are known as the targeted site. And in the target site we have the receptors which tend to receive the hormones, and then we have different mechanisms in which the hormones they exert their effect. Now in plant cells it is different, because sometimes the hormones can be produced by the certain types of cell, and they can exert effect on the same kind of cell at another place of the plant, or even at the same site, or even nearer to the place of production. Na ndio maana nikasema kwamba among the mechanisms ambazo phytohormones zinakuwa zinafanya kazi au zinaweza zikawa zinafanywa translocation, inaweza ikafanyika hata cytoplasmic streaming. So cytoplasmic streaming and passage of phytohormones through plasmodesmata from one plant cell to another plant cell inaweza katokea na hormone au phytohormone na move kutoka kwenye one cell kwenda kwenye another cell. This is different from the animal hormones kwa sababu zenyewe lazima zipite through the circulation system. And another characteristics, hormones or phytohormones they are transported within the plant by utilizing 4 types of movement, which are localized movement, cytoplasmic streaming, vascular tissue. They are used to move hormones, as how we shall discuss later, because for every phytohormone which we will discuss we need to know about its site of synthesis and how does it distributed within the plant body. So the distribution of each kind of phytohormone we'll discuss it later, and the site of production. And then the last characteristics, we said the production of hormones occur very often at sites of active growth within the meristem. So for most of the plant hormones which we discuss, or phytohormones which we discuss, most of them they are produced at the active site of growth, which are meristematic tissues. And this is because these substances which are called phytohormones, they are sometimes called as plant growth substances. So they exert their effect most often it is by either stimulating the processes of growth in plants, as we said that cell division in plants, growth in plant can involve 3 processes, which is cell division, cell elongation and cell specialization. So these hormones they tend to do their effect in either stimulating or inhibiting these processes. And basing on the groups of phytohormones, we have the 5 types of phytohormones which we will be discussing in plant coordination. First we have auxins, then you have gibberellins, we have cytokines, abscisic acid and ethine. Among these, some of them they tend to stimulate growth, and some of them they tend to inhibit growth. So their effect can be either synergism or can be antagonism. Now when we are talking of synergism, it means that the hormones they are working together to perform a certain function. For example, gibberellins they can stimulate cell elongation in the presence of auxin. So gibberellins themselves they can't stimulate cell elongation, but in the presence of auxin they stimulate cell elongation. That means gibberellins tend to support or tend to do its function in the presence of gibberellins or by support of gibberellins. Also, even cytokines, they perform their function in presence of auxins. Also, in the case of abscisic acid, abscisic acid tend to do antagonism of most of with the effect of most of the hormones, and more especially auxin and gibberellins and cytokines. Abscisic acid tend to inhibit the processes which are stimulated by these hormones, and by that way, abscisic acid, its working mechanism is more of antagonism as compared to the working mechanism of other hormones. Now, let's start our discussion by discussing auxins. Now, auxins, these are major plant growth promoting phytohormones. So, if we are discussing about the plant growth substances, don't forget about auxins because they are very important. And if you understand on auxins, it will be very easier for you to understand on other phytohormones how are they working, because as I told you that most of the phytohormones, and more especially gibberellins and cytokines, they are working in the presence of auxin. Now we have several types of auxins, because auxin, when we are talking of auxin, we don't talk of one compound. It is the group of closely related compounds. Now we have several types of auxin, but the common one is the indole acetic acid, IAA, the indole acetic acid. And you need to know some of the aspects in these phytohormones. As I told you that in every phytohormone you need to know first about the basic of that phytohormone, then you need to know the discovery, how the scientists they did the experiment, or what factor they lead them to discover that phytohormone, and then later you need to know it is synthesis and the distribution. And after knowing the synthesis and the distribution, you need to know about the laws of that phytohormone in the plant, and then finally you need to know about its financial or its commercial application. How does it use the industrially or commercially? Now in the case of auxin, let us move to the discovery of auxin. As I told you, auxin is the group of closely related compounds. The most common one is the indole acetic acid. So as you can see in this chemical structure, this is the indole 3 acetic acid, and we have this basic ring structure, which is indole, composed of benzene ring and the cyclopentene ring. So benzene ring and cyclopentene ring together they form an indole, and this is the basic structure of indole. So as you can see, we have the indole 3 acetic acid, we have indole 3 butyric acid, we have indole 3 acetaminidine, we have indole 3 acetaldehyde. But as you can see here, the difference is the side chain, which is attached to the parent chain, which is indole, or which is attached to the permanent group, or the we can call it the flame, to which every kind of phytohormones have this kind of skeleton. So every kind of auxin must have an indole skeleton, and then it has the attached group. Now in the case of discovery of auxin, these are just chemical structures of auxin. We have the indole skeleton, and then you have the attached group. In the case of discovery of auxin, they did a series of experiments, and based on the results of those experiments, they become knowing that this probably there is a certain chemical substance which tend to influence these kinds of growth. So the discovery of auxin was the result of an investigation into phototropism that began with the experiments of Charles Darwin and his son Francis using oat coleoptile. Kwa hiyo discovery ya auxin ilianzia kwa kutumia oat coleoptile. They showed that growth of shoot towards light was a result of some influences being transmitted from the shoot tip to the region of growth behind it. Kwa Charles Darwin pamoja na mtoto wake walichogundua ni kwamba the growth of coleoptile depends on a certain chemical substance which is probably produced on the coleoptile, kwa sababu they didn't have devices to prove that, but according to the experiment ambazo walifanya kama Charles Darwin na mtoto wake with the other scientists, now they become knowing that probably there is a certain chemical substance which is produced at the coleoptile tip, and it is distributed to the region of cell elongation or cell division behind the coleoptile tip, and these growth substance tend to influence the multiplication of the meristematic tissues and their elongation. As we said that the auxin, in presence of other hormones, they tend to stimulate cell elongation. So here below we have the table, or we tend to show experiments done by different scientists, and these experiments later they made the scientists to know about the auxins, their properties, and how are they functioning in the plants. So in this video, let I end up here. In the next video, I will start explaining about this experiment which led to the discovery of auxins, and then later we'll move to the rules of auxin and their commercial application. Thank you everybody. Let I wish you nice studies. Don't forget to subscribe and to like this video.